Okay, so thank you so much first for the invitation, Noam, Amy, and Carolina. It was a really great day already. So today I will tell you a bit of a story of how someone that does imaging or starts as an imaging lab ends up with structural biology and AlphaFold. And maybe some of the tools that I will present today are also useful for the other people here in the field that are doing imaging. So in our lab, we are interested in herpes viruses, and I don't have to introduce them again because there have been great interactions before. And so the first thing we are interested in is morphogenesis. So that's basically how virus particles are coming together and how do they, for example, leave the nucleus, as you can see here. Is there a pointer somewhere, or is this built in here? Is there a pointer somewhere? The white button? There's no white button. Well, doesn't matter. So one of the things we're interested in is how virus particles actually exit the nucleus. So with herpes viruses, capsids are made, and they have to leave the nucleus, and the nuclear pores are too big for that. So how do they get out of the nucleus? That was one of the questions that the field has looked at for a long time. And so the process that is looking like this. So you have basically the viral particle that aligns with the nuclear membrane, the inner nuclear membrane. There's this neck, so the nuclear egress complex built. That leads to scission, and then you have this prime envelope particle in the middle between the two nuclear membranes, and then this particle fuses and is released. This process is studied ad nauseam in the herpes virus field. However, nobody has looked at this in living cells. Now, we took the structured data that is available and generated a lifestyle assay. And so we used this data to really find the right combination of tags so that this is working. And this is now cryo-ET data together with Kai Grünewald's lab, where we actually can really target single particles here that are just enveloping at the inner nuclear membrane. And you can find them then in cryo-ET, and you can also see here nicely that they're enveloped. So our assay works. And this is now how it looks in living cells. So what you can see is virus particles that are here enveloping. So you see that these dots are coming up. You also see dots disappearing. So this is basically the event where the particles are getting outside. And excitingly, you also see these particles here that are enveloped, and then they're actually moving actively in the nucleus. And this is something that is completely new. And I think this is also the power of lifestyle imaging, even with these processes that have been studied for 30 years, that suddenly you see things, how they really happen in the cell, right? Now, why am I telling you all of this? This basically should be the introduction of why structural biology or structural data is important to make our mutants, to make functional assays. And ideally, you have that structural data already, and that's all easy. But most of the time, you have a question, and then you want to do the functional lifestyle assay. And this, by the way, is ChatGPT of how single particle tracking should look like. But there's the step in the middle here. And that step is this annoying structural biologist that takes 3 years or even longer to make a structure, while you just want the interface, right? You just want to know the interface so that you can break it and you can look at it in your functional lifestyle assay. So the rest of my talk will be about cutting out the middleman. And I know it will annoy a lot of people here. We are also doing structural biology. It's not that bad. Now, this was basically the time when AlphaFold 2 came up. So the first thing we did was we predicted all the human herpes virus proteomes. We clustered them into groups. This is all on herpesfolds.org. This is also really nice and interactive. So if you're already bored by now, go to the website. You can play around there. It's really good. And just by predicting the single structures, we already find some new families. For example, this one protein is one of our pet proteins, Hul 112, 113. It phase separates. It's important for viral replication. And it also forms filaments. And this is a 3 angstrom structure or envelope of exactly that protein. So we're also doing some structural biology. And we also found that HSV, for example, that was introduced earlier, also is doing that. So now you can, with the structured data, you can build assays around that and do that. But as I said, most of the time you need an interface, right? And that was the second step, and this is called herpes PPI. So what we did here is, and this is now really work that mostly Tim did in the lab, he's a very talented postdoc. He took basically the proteomes of the human herpes viruses and predicted every protein against each other protein. So that's about 25,000 predictions for 4 different viruses, or 10,000 GPU hours. And so that gives us a long list of predictions, and when we basically scored them by IPTM, we used a yeast-to-hybrid data set to figure out a lower threshold for the IPTM. I can go into detail in a personal discussion if you like. And then basically determine the cutoff. Now, all this data is on our website that you can see here. So this is herpes PPIs. And for example, for HSV, it gives you this predicted interaction network over the proteins. And if you click on it, it opens another window. You have the structure, you have the interface. Now, you can do this for different viruses. And the cool thing is with this the interface, you can now click on several buttons at once, and that gives you the conserved protein-protein interactions, the predicted conserved protein interactions. If you now take this data and compare that to what is known in the field, and that's like 50 years of herpes virus research, it almost maps 100%, basically telling us that AlphaFold can recapitulate a lot of the research we've done before and figure out the conserved interactions. That was, for me, a point where I really thought, okay, this is powerful, right? Now, this is also kind of boring because, as I said, this is known. So what about all the other things which are unknown? And this is basically all this. So this is a list of the high confidence interactions of HSV1. And you can see here the ones which are described, and all the blue ones, they are unknown. They haven't been described before in the literature. And this is basically just tediously going through the literature and also using Interact to figure out that list. Now the question, of course, is how many of them are real, right? And you can see here, they go from a very high IPTM to lower IPTMs, and you can see most of the unknowns in the lower part. So how many of them are really real? Now, we could go back to our structural biology friends and do structures of all of them, but that will take forever, right? You could also try to express all of them and use them in kind of an interaction assay, but again, that will take forever. If you have ever produced a protein, I'm not a biochemist by training, but it takes a lot of time. So we figured out a way of how to do it, hopefully. We call this Lucia. So the basic idea was that we take one protein, the A protein, and we put a his tag on it. There's also an alpha tag on it for titration purposes. And then we have the B protein that is supposed to interact with this, given the prediction. And we put a nano-look on that. And then we have to express them. And that is the bottleneck, as I explained, right? So basically, Yulia and Tianzhu, they figured out a way of how to do this completely in vitro. So there's no E. coli involved anymore. We can go now just by PCR. That PCR is then hi-fi assembled to the minimal backbone. This is basically just closing the whole thing into a plasmid with a few enhancer sequences in there. Then we do a rolling circle amplification step. This is also in vitro. And at the end of the 2 days, we have our protein because we express it in the cell-free expression system. Now, the amounts in there are very, very little, but we only need very, very little. So this is like in a 10 microliter reaction now, and that can be then optimized and done in high throughput. Now we take our protein now, the A protein, and stick it on a hist-coated plate. And then we put our B protein on top, we wash, and afterwards we measure our fluorescence, the luminescence output in the luminescence reader. Now, because this is all in vitro, this goes very, very quickly. As I said, in about 2 days we can do that. So we have done this now for HSV1. And these are our controls here. So we have a protein that is MBP alpha tag that's sticking on the plate. And then just nanoluciferase as a B protein that gives us a very low fluorescent luminescence. Now, there's a minus and a plus, and if you want to know the details of all the controls we are doing and how we are actually normalizing this, we can talk about this later. The positive control is an MBP alpha tag plus a nanobody against the alpha tag with a nanoluciferase on that, and that gives us a very high signal. And you can also see this here, so the IPTM for this interaction is very high, very low for this one, and this is also then correlating with the luminescence readout. Now, what about known direct PPIs? You can see them now here. So these are all the black ones. They're all positive, except one, and that is a dimer, a direct dimer. So I think we are unsensitive to dimers, so basically two A proteins can bind to each other, so that's why this is not working, if it's a homodimer. But the rest is working. And if you now go to the novel direct PPIs, you can see that about 60% of the novel PPIs we can actually confirm in Lucia, and about 36% of them we can't. If you now look at this by RPTM range, and maybe you know AlphaFold, and DeepMind basically says everything from point A to 1, that's the high fidelity zone. These are interactions that are likely to be true, and that's what we're seeing. So all the interactions that are high IPTM, we actually can confirm. And everything between 60 and 80%, DeepMind is saying this is the gray zone, so this is where you have to, we can't be sure. And what we see here is that we see about 64% of them are positive, right, in our assay. So I think this is very good because it basically confirms what DeepMind also tells us, but I think this is the first time that someone has done this experimentally in an interaction assay. And then, of course, we have the low confidence PPIs, and they are clustering low. Right, so we have now confirmed interactions, Lucia confirmed interactions, but we want the interface, right? That is the important thing. Now, the thing is here, this is a prediction. This is not another yeast-2 hybrid that just gives you binary yes or no, right? We have the predicted interfaces. So can we now actually confirm them? So for that, we built a pipeline of now to extract the most important amino acids in this interface, in the predicted interface. We call this B-scan. And Ingmar was the leading force on this. And for example, for this interaction, this is UL8 with UL42. This is a novel interaction. So this is the DNA sliding clamp. This is part of the helicase complex. We have this predicted interface here. We identified the most important residues. If you now predict mutants that have these residues mutated with AlphaFold, you can see that IPTM is dropping. And if you combine 5 of them together, we also see a nice drop in our luminescence Lucia assay. So basically telling us that in general the system is working, so we can go from prediction to large-scale predictions to narrowing down to the interface, mutate the interface residues, and tell us, okay, these are the interface residues that are important. With this, I'm at the end. I think this will be useful for a lot of you that do imaging, because now you can basically go to our website. There will be an updated version with the mutated interfaces. You can use this technology now to basically make functional assays and look at this in living cells. With this, I'm at the end. I would like to thank my group, who really did all the work, you for your attention, and the funding, of course. Thank you.